नाज़रीन, कश्मीर लाइफ के साइंस टॉक सीरीज में आपका खुशामदीद। जब पूरी दुनिया कोविड के खिलाफ लड़ रही थी, सब लेबोरेटरीज, सब साइंटिफिक इंस्टीट्यूशंस पूरी दुनिया में इस वैक्सीन की तलाश में थे, तब एक इंस्टीट्यूशन का जिक्र रोज बरोज आता था खबरों में। उसका नाम है FDA। ये बेसिकली अमेरिका की सरकार का जो डिपार्टमेंट है ड्रग्स और फूड का, तो इस हवाले से दुनिया का सबसे बड़ा इंस्टीट्यूशन है। तो यूजुअली जब कोई ड्रग बनती है या कोई मेडिकल इक्विपमेंट बनता है तो उसको बड़ा एजाज होता है जब FDA में उसकी अप्रूवल आ जाए। तो आज की निशस्त में साइंस टॉक्स की सीरीज में हमने FDA से एक की डिपार्टमेंट के डायरेक्टर डॉक्टर हीरा लाल नखासी साहब को दावत दी है, जो इस वक्त हमारे साथ वाशिंगटन से हमने राब्ता किया है। आज इस वक्त वहाँ सुबह है तो यहाँ शाम है। डॉक्टर साहब, आपका खुशामदीद कश्मीर लाइफ के शो में।
Thank you, Mr. Hussain. I really appreciate the opportunity to talk to you, and I'm really honored to be asked to, as a Kashmiri, to talk about what we are doing, about my journey and my science. Thank you.
Dr. Sahab, just for a start, we are not talking to a scientific community, we talk to commoners. How can we understand what FDA is all about and what does it do? What is the long, vast interior of belly of FDA and how it is so crucial to the humanity on Earth?
It is very important because FDA is sure that's a regulatory body, and any drug or any biologics or any device, as you mentioned, Mr. Hussain, has to be evaluated. You cannot sell anything by just making in your garage or in your factory and sell, because you need to make sure Lukan Gastarun Icha safe is Icha Karakam Chakaran na agar ka vaccine right or ka drug hai, so does it work? So the FDA's role is to make sure it is safe and efficacious. Means it is safe, you can get a problem, or it will cure you from that particular disease. So that is the main mantra of the FDA, because in the FDA we regulate biologics such as blood problem. So you need to get the blood transfused. You want to make sure you blood transfuse gets look and months. That is clean. That's an infection diseases can. Similarly, vaccine, you should say like the COVID vaccine. COVID vaccine, you must point, it's not, something like, you don't get a placebo effect. Same thing, organ tissues transplantation. You want to make sure those organs are clean and free of infection. So that is a major responsibility of the FDA.
डॉक्टर साहब, इससे पहले कि हम आपके साइंटिफिक परस्यूट्स के हवाले से बात करें या कोविड-19 की जो सूरत हाल अमेरिका और इससे बाहर रही उस पर बात करें, ये जानना जरूरी है। नाजरीन भी ये जानना चाहेंगे कि डॉक्टर साहब का जो सफर रहा, लर्निंग कर्व रही, तो ये कर्व बहुत चैलेंजिंग रही होगी, क्योंकि कोई चीज देयर आर नो फ्री लंच। सो तो ये जानना चाहेंगे कि आपकी जिंदगी के बारे में और आपकी स्ट्रगल के बारे में, और उसमें जो लर्निंग कर्व जो रही उसमें जो बैंड्स आए होंगे, उनमें कितने श्रीनगर जम्मू शाहरा के बैंड्स थे और कितने जलेबी मोड़ थे?
Thank you, Mr. Hussain. So yes, Kashmanan, as I mentioned you off the camera, I was born and brought up in Kashmir, Ranawari, Srinagar. Actually, I was when I was in my high school, I studied in Sogam Lolab. You may be knowing Sogam Lolab. Yes, because my dad was a physician, so he was posted there. So I had to, because we were small, so I graduated high school from Sogam High School. So from there, I came to Srinagar. But I was born in Srinagar, so Ranawari. So there I went to Gandhi Memorial College and SP College, one at SP College, but at Gandhi Memorial College and it was struggle because I wanted to, my dad wanted, actually, not me, dad wanted to go to medical school because he was, and I was not interested, but I tried and I just didn't get it. Anyway, so I got a little bit distracted in there. But then as, I wanted to go out and explore what is more there because, in Kashmir, we, at that time, I'm talking about in early 70s, Scott, there were not many options there. So yeah, it was medical school, yeah, it was engineering college, and there were only limited seats, so everybody couldn't get it there. So, but there was nothing known. There are other disciplines such as biochemistry, physics, mathematics, where you can also yes, excel. So I had a friend who was, living close to our neighborhood, in our neighborhood, Ranawari, and his brother was doing his PhD in biochemistry in Baroda. And I had not heard Baroda Kachu Kyachu Jai Bront before that, because I had never gone out of Srinagar, to be honest with you. So yes, so I applied. I got into the biochemistry department of Baroda, MS unit. It's called Maharaja Sayaji Rao University, Baroda. And I excelled. I did both masters and I got first division in there. Then my professor said, you want to do PhD because you are good at science. And, I said, okay, because, I didn't know better than that. But I thought I got interested in research at that point because he was his name was Professor C.V. Ramakrishnan, very, very, knowledgeable and respected professor at that time. So I took his advice, did his PhD, and he was so pleased. He asked me to go to US for 6 months in Ohio State University to get more training, because the training which it was very limited those days. So now it is different. So I went. I was really glad that it opened my eyes how research is done, not just added this and that, and which I had learned there. So I came back, finished my PhD, and I requested him that I will now go back to US because I don't feel I am complete with my research. If I have to pursue my research career, I have to explore more. So I came here in NIH, the National Institute of Health, which is a premier body of the US, which does conduct research. And I was there within National Cancer Institute. So I did some work on cancer biology. Then within the 2 years, I said I need to get more training. So I went to Columbia University, which is in New York, which is another Ivy League university in US, a top-notch university. So I got there and did further studies on the cancer, liver cancer there. Here in NCI, I was doing the breast cancer. But during that process, I learned the new field which was coming up, molecular biology, which was not in that time. Nobody knew. So I learned the techniques. And after finishing my postdoctoral training in Columbia University, I got a job in NIH again. But then within a year or two, I moved to Food and Drug Administration. This is in 1984. So then I was given independent lab to work on the projects which FDA wanted. First, I worked on the rubella virus vaccine, which had some problems. They asked me, because I know molecular biology, can you find out the problems? So we did, and it was published. And so then came along a lot of twists and turns in my life. So, moving from one field to another field. So this was virology. So I learned virology because I was cancer biology. So I learned virology. And thanks to colleagues who were in that time, they taught me about how to grow the viruses and all this. Then my boss, who was at that time division director of our division, he said, we have a bigger problem. I said, what is that? There is this parasitic disease which causes lesions in the body, and also if infects people, it kills people. There's medicine, but it doesn't do much, So, and then I read about it, and this disease is very common in India. Actually, this disease called Leishmaniasis is common in eastern UP, Bangladesh, West Bengal. Major epidemic center is in Bihar and Nepal. And now it has spread into other. I heard recently there are cases in Jammu and these cases, and Himachal Pradesh, and Rajasthan, Kerala. So he suggested that why don't you work on this thing? So again, I had to learn new things because I had not seen, did anything on the parasites. So luckily, NIH is a place where you get world's experts on every subject. So I went and talked to a few people. They were very open and giving. They said, no problem, come and we'll show you how to culture the parasites. But the rest, I had ideas how to do it. So I started with that, which was in almost 1990. So I started working, and immediately I realized that there is no vaccine for this thing. So I need to develop a vaccine which will cure these people. So that time, a lot of people, it was not that people were not trying, but there were a lot of people trying different techniques, just like the COVID, and they were taking a little bit of protein, adjuvant lagavan, or it's a virus, to inactivate corruptive. But all these things didn't very well work in the case of leishmania. But what worked in the past, agar Iran, when they used to have the Iran, this disease is very prevalent. Iran-Iraq war is. So that time, a lot of these Iranian soldiers got this infection because Iraq is an epicenter as well as Iran is. So my colleague, his name is Ali Kamishpur, very nice person. He's from Iran, scientist, trained in US. He had done studies where he would take a little bit of this virus, parasite from a skin and put onto kanas body but it was viral wild type, just but a small amount. So a lesion but it will heal itself, but people were getting protected. So it gave me the idea that if you have a parasite which is not virulent, which doesn't cause this lesion, small amounts of lesion, but can protect against the infection, why can't we do that? So me, my colleagues, Dr. Abhay Satoskar from Ohio State University, Dr. Greg Metlishevsky from Canada, Dr. Shinjiro Hamano from Japan, and we got together. These are my colleagues who work in this field. We got together. I said, this is a concept. Agar asa parasitus gene, agar ye remove karo, which is causing the disease, can it be less virulent or can it not cause disease, but it will protect? So it was, novel idea at that point. And so we did, luckily, and with this, our colleagues. So we used this technology called CRISPR-Cas, which is now, you must have heard a lot of this thing where this is, you remove the genes from a cell and so that like cancer patient or blood cells, they remove the cancer gene and then put back this thing. So using this CRISPR-Cas technology, we then developed this vaccine candidate, and actually several of them, 2 or 3, but one candidate was, we tested them in animals because you have to do when you want to go through the approval process, FDA, you have to do preclinical, means animal model and to are safe and efficacious. And then you have to do in humans as a safety, and then finally. So we made that thing, and luckily we got funding from Japan, we got funding from NIH, we got funding from Wellcome Trust because they saw this, potential for this thing, this vaccine candidate being manufactured in India, in Pune, a company called Genova. And Dr. Singh, who is the CEO of the company, was a friend of mine when he was in NIH. And he usually, Kashmir, this kind of disease is poor man's disease. Big companies like Merck, Pfizer, or like, who all develop COVID things, they don't touch it. Because I talked to them, they said, oh, there is no profit. Garib logan country, you have to sell it and then lost price kind. So nobody is really interested. But this guy took a challenge because he was from India. He is from India. He's settled, and it is disease in India. And yes, so he said, let me help you guys. And so he is manufactured now under the good manufacturing conditions, which is called GMP conditions. When you make a vaccine, you want to make sure it shouldn't cause a problem. So therefore, and now they have done that. And inshallah, we will be ready to put into people next year, beginning this month. So to look at the sick. So this is my story, my life story starting from Kashmir, Ranawari to where I am now.
It is very impressive, very impressive. Let us go back to your doctorate. PhD किया तो अब आप किस पे काम कर रहे थे, and what were the takeaways from that? And
PhD level, yes, Kashmir, my boss had suggested in a project, and which I took upon. In Kashmir, Hindustan is a lot of malnutrition. Because in those days, especially now, I think it is more of obesity. So a lot of malnutrition, even still in poor places, people don't get enough food, and they are protein deficient, carbohydrate deficient, fat deficient. And my boss had, and his wife had done studies before, in people who are these malnourished, their cognitive thinking is low. Poor guys can get susceptible to all diseases. So he said to me, can you find out what is happening in their brain lipids, brain develop? Because what happens is, when you have not enough food, when you are born, your brain development is affected very much, because brain is the first year and a half. The whole development takes place. After that, that's it. So we use, not in the people, I couldn't do these experiments in the people. So I had to do in rat, a model, where we artificially created malnutrition. There were normal food given, in some cases, no protein given, no carbohydrate given, no lipid given, and asked the question, what is the brain development happening? Is it affected? And it did turn out to be that brain is a sheet called myelin sheet, which covers the brain tissue. That is affected. Cognitives are not very well. So because of that, because he saw the data, my boss, that's when I told you that he asked me to go further training in Ohio State University, where they were already doing that kind of with a nicer, much more refined techniques. So I learned lipid measure, protein measure, but now Yes, in the Ohio State University, Columbus, Ohio, I got the opportunity to learn new techniques to go a little bit deeper into it. That excited me to come back and to do more research on other things.
जब आप finally वापस चले गए अमेरिका और settle हो गए, तो other than the vaccine you talked about poor man's disease के हवाले से, तो in between what were the key projects that you handled?
30 years. This has been my major focus. And this is because you cannot do some side projects or anything. It has to be concentrated. And so developing vaccine takes years. We were lucky that in the COVID vaccine, because of this new mRNA technology, and it worked. Nobody knew it will work. And also the other technology, we could get it in a year's time. But usually, and also if you remember, these are emergency use authorization. They're not fully approved yet. So 1 or 2 may be approved fully, but most of them are emergency. Means that agar kya problem with safety or something, They will take the authorization from there. So any vaccine which you have to develop, it takes a long time and dedication of a lot of people. And in my lab, I want to thank those people who worked with me over the years. There were, almost 10, 15 people who went through the lab and contributed. I'm not the only guy. I was just directing them. Besides that, and I told you one project which when I joined FDA was this vaccine, rubella vaccine, which measles, mumps, and rubella, which every child gets these days. I don't know whether rubella you get in India or not. Here, everybody gets it. So when the rubella virus vaccine was developed, they found out that you look in the people who got this vaccine, they started developing joint pains. So the question was, what was the problem that vaccine was? Some people, not everybody got the joint, but there were a significant number of these adverse events which causing the joint pain. And also, if by mistake a pregnant woman got this vaccine, it affected the child. So I was tasked, what is the problem with this vaccine? Can you figure it out? And then the vaccine, companies can modify this and then change it. So what I did was, knowing I was a molecular biologist, I knew all the molecular biology tool. I sequenced the whole genome, which is around 10,000 bases. And so we found out that there were few 10, 15 changes in the vaccine, which would cause this disease, which would cause the knee problems or arthritis. Basically, it was arthritis. So then with that knowledge, we gave back to industry to modify it. And obviously, they did that and that. So that was another major project. And when I was in NIH, I did this breast cancer projects in the mice. Basically, you couldn't do that, and find out there were certain milk proteins which are important for the development of the breast. And if there is a change in those expression of those milk proteins, then some of these cells which are producing this milk in the mammary gland, because there are cells that can become abnormal and cause the disease. So those were the other two major. But my life's focus, which is the major focus, has been this understanding, first of all, not only vaccine, but first of all, understanding how this parasite Leishmania causes the disease. We also worked a little bit in collaboration with other people on malaria. Malaria vaccines. There are colleagues in my division who work on malaria vaccines, and it's again, that parasite's a different beast. And people have been trying to get the malaria vaccine for the last I don't know how many years, and and the drugs when they take that, they causes resistance. So that part I'm not fully engaged, but I contribute time to time. But my major focus, my focus for the last 30 years has been because I saw in my head where I can go with this thing, and the vision, touch wood, I hope it becomes successful. And
what is your current role? You are heading a major division in FDA.
My day job and my regular job. My day job, which is an administrative job, which is I am head of the division called Division of Emerging Transfusion Transmitted Diseases. Because any blood in this country, in the United States, when it is transfused into people, those who need transfusion, so it has to be tested against various agents which are transmissible through blood, like HIV, hepatitis B, hepatitis C, West Nile virus, Zika virus, Chagas parasite, Babesia is another parasite. So the manufacturers make these test kits. These are test kits to measure to test these blood samples before it gets transfused into people, to test that whether it doesn't have any of these infections. So my division is make sure that they evaluate all those devices which are used for blood donor screening. And there is a people, almost 100 people in the division, who really look at all these applications for the devices which come to my division. And because it's a major responsibility to ensure the United States blood safety and availability. So then there are also in the division, which I oversee them, they do this research, just like my lab is doing on this Leishmania infection, vaccine. Same thing, malaria vaccine. Same thing, HIV vaccine. Some HIV. There are at least 10 investigators. Means we call it independent investigators who work on these different projects, and they also review these applications.
Normally, जो हकीकत भी है और जो perception भी है world over that America is जो knowledge का pyramid है. तो given the strong institutions built over centuries, लेकिन चाहे साइंस की बात ले लें या किसी और फील्ड की बात ले लें, but when COVID-19 came, in comparison to many other countries, initially it didn't respond so well, or that is the impression. So what could be the reasons कि जहाँ सबसे ज़्यादा knowledge थी, सबसे ज़्यादा एहतियाती institutions थे, that took a huge toss.
Yeah. Unfortunately, in this game, in these situations, there's a political situation and then a scientific situation. The political was that they thought it is not, it's somewhere in China and this and da. But when it started coming in the US, then people got scared. So that's when the government immediately put a lot of money into the industry. But I think in the beginning, unfortunately, it's my personal opinion, that people did not realize the potential of this disease. They say, it will maybe in zukam, sugar, flu type thing. But when people started dying, that's when it hit. It was started in March, kind of 2020. First cases started come February, March. I think I was in India. I came back on February, and right after that, I heard, there's these cases in Seattle, Washington, and and then it just, and they shut down everything from, I think I remember March 16th was that we could not go to the lab, we could not go anywhere because they wanted to stop the spread. But the vaccine manufacturers, they were given a lot of money by the government to develop, and which they did in a very fast. It's the only thing is not that they did the fast, but then we have to approve. It's just and there was a lot of pressure from the political leaders. Hey, fatapat approve. But I want to give credit to my center director, my head boss, who said, I don't want to cut corners. I want to make sure that it is safe, because it will go to millions of people. And we are the beacon of the regulation throughout the world. As you mentioned, we cannot afford to have something as a second class and which causes diseases or will not protect to go out of on the market. So he was very much under pressure, but we all were under pressure. We can't, even though I was not directly involved in reviewing them, but I took because those people who were reviewing, their workload was their original, regular work is theirs, that cannot go. So many of us took their workload so that the rest of the work keeps on going. When people get the infection, then they develop antibodies. So their blood contains a lot of antibodies against the COVID. So we were asked, can we approve those blood, plasma, basically. It is the, source plasma. You collect the blood and put the cells back, and then they take the, liquid thing. Can we look at the titers of these antibodies and then inject into people? Because that was one of the therapies also initially to treat people, because the vaccine also, there was no drugs. So this was the only thing. If you could take antibodies from the Christians antibodies and inject it so that it can subside the infection. So that we were very much busy with that. And then came the monoclonal antibodies, and then came the vaccine. So once vaccine came, so everything fell out. I think that was good to have that. I'm really glad.
While fighting COVID, you told the story about what was the response from world's most powerful democracy.
Correct.
Was it the same case in world's most populous democracy? I'm talking about.
I think that you're right. That's a good question, because in the beginning they thought, it is outside, and they didn't get that many infections, or maybe they didn't notice that. But when in 2021 January, July, and when you saw world over saw that bodies being cremated throughout on TVs, that jolted everybody. Say, what the hell is going on? And that is when the vaccine, which was developed by AstraZeneca and manufactured by Serum Institute, Modi then told them that, look, you got to give that vaccine to people in this country. I was surprised. Within 6 months or so, 1.4 billion people, 1.3 billion people were vaccinated. And that was otherwise, I was very much worried. Look, I will tell you my personal story. My brother passed away. So my younger brother with COVID in Jammu. And, so this is really sad stories. It has touched every family. And, my cousin, one of the cousins also, my first cousin, my Pitur boy, poor guy in Himachal, he got somewhere, he passed away. So these are young guys. So I was getting very worried, because at that time there was no vaccine. Just put the mask, keep the social distance. You can't vaccine guys. But they say, it is nice. And I got so upset with my brother when he went to the hospital. I said, what happened? How did you get it? And poor guy, but after 10 days, he passed away. So, he just, it's very sad. Any largest democracy in the world, or largest populous democracy in the world. And, other countries shut down. And then when they hit them hard, both a million people died here, and I don't know how many million people have died in India. They realized that they have to take these draconian, just like China did. And, they just try to, I don't know how many people they really have died, but, we do not know that. But they took those draconian measures to shut everything down. Yeah, economy can affect, but if people are not there, what good is economy?
It has been a very impressive, knowledgeable conversation with you, जो आपने हमारे साथ की. अपने बिजी शेड्यूल में से टाइम निकाल. We are so grateful.
Thank you, Masood Saab. It's my honor to talk to you, and I'm really proud to be a Kashmiri, and also, if I could contribute to the spread of knowledge in my friends and colleagues in Kashmir, or younger generation in Kashmir, if once they listen to it, there is, they should be, there's a hope. And I want to keep that hope alive. And there is a better future for people if they don't find opportunities. I would recommend all these young kids, if there is, I don't know what the situation in institutions in Kashmir is, but there is better institutions and much better infrastructure in the rest of the country. They can, or in the US, or in other Europe or places. And so I think there's a better future for that. If anybody needs any information, they can send on my email. I can direct them to.
Thank you, sir. So nice of you.
Thank you.